Hello students, this is Dr. Arindam on behalf of Hippocampus by Medeasy Tutorials, and I am here to discuss the questions of Aiims PG June 2020 exam. Alright, so all total there were 9 questions in biochemistry, so I'll right away get in the point. So the first question from biochemistry was identify the compound given in the image. So it was an image based question. So you can see the image over here, and the options were keratin sulfate, dermatan sulfate, heparin and chondroitin sulfate. Now this question is actually very easy for those who know in detail all the structures. Even if you do not remember the structure, if you know the constituent formulae, then also it will make it easy. In specifically this image, this one is glucuronic acid and this one is galactosamine. Now there is only one combination of these two, alright, repeating units of glucuronic acid and galactosamine is nothing but chondroitin sulfate. So that's the answer to this question. Moving on, next it was again an image based question, and it was one of the tough questions from biochemistry this year. The question was very simple. This diagram was given and it was asked which of the following is fetal hemoglobin, right? And below the diagram a legend was given, which one is alpha hemoglobin and which one is beta hemoglobin, that class. Basically what is being asked here is you can see part in the total synthesis of globin percentages. All right, so we all know that alpha hemoglobin is that the one in the top blue is always synthesized, starts in the embryonic stage and it goes on and on and on. I'm coming to the explanation, but the answer of this, so option were either 1 or 2 or 3 or 4. So the 1, that is the first blue curve that goes up like hyperbola and continues. Number 2, the one that starts developing in 6 weeks and then continues to develop from 12 to 24 weeks and then generally wanes off after following birth. All right, the next are 2 lines side by side. Those are combined, option 3. They start in embryonic stage just and then ceases to exist in between 6 to 12 weeks. And fourthly, the one that starts developing slowly, it starts around after 6 weeks, slowly rises and then rises after birth and then continues to take off and continues to rise and become a high level on the around 18 weeks after birth. So as per your logic, what does it say? Even if I don't know what's the answer. So let's analyze graph number 1. So if it was fetal hemoglobin, it's starting from 6 weeks embryonic stage and it is continuing at a high level lifelong. Does it happen? It does not. Considering number 3, okay, if this was fetal hemoglobin, it is already becoming 0 at between 6 to 12 weeks. So it can't be, because we know fetal hemoglobin plays a very important role in utero as well as few weeks after birth, right? If this is number 4, so what is actually number 4? Number 4, it starts to develop, it is low in intrauterine period, it is not so high at birth, almost 30%, and then it slowly rises and it continues to remain at the top. So it is actually adult hemoglobin. All right, and what's left? Yes, number 2. If you chose it from your common sense, you got the right answer. So fetal hemoglobin actually starts to be synthesized around 6 weeks of life. After all the other embryonic hemoglobin, that is epsilon hemoglobin, alpha hemoglobin has already played their major role in oxygen carrying. It is the major hemoglobin that is in a peak level in utero, and then it falls down slowly. So see, at birth it is in a much higher concentration than the adult hemoglobin, and then it falls. All right, so this is the reference diagram. Okay, this is taken from this journal, British Medical Journal, and this is the full diagram. Okay, the upper part was smartly truncated in the Aiims exam, So it is actually during the yolk sac in the early stages of life. Okay, alpha, delta, gamma, epsilon. There are eta. There are multiple types of hemoglobin that are formed. So starting with yolk sac, and then liver takes over, and then spleen, and ultimately bone marrow. So megaloblast, macrocyte, and ultimately becomes normocyte, the RBC. So as we have guessed it right, the alpha gamma, we are talking about gamma because that is the main globin part of the fetal hemoglobin, and so your answer rightly is option number 2. Okay, moving on to the next question. See, the next question was a straightforward question. Which of the following is not an enzyme in cholesterol biosynthesis? And your options are HMG-CoA reductase, HMG-CoA lyase, HMG-CoA synthase, and thiolase. Now let us look at the cholesterol biosynthesis. So as you can see, and I think you must know that the enzyme thiolase, HMG-CoA synthase, and HMG-CoA reductase, okay, just you can see the first top 3 steps, okay, they are very much part of the cholesterol biosynthesis. Okay, even if you are preparing for your undergraduate lessons, we are often told that please read up to mevalonate, okay, because the later part of the synthesis is not that important. Okay, even then you can answer this question. And moreover, this enzyme HMG-CoA lyase, it is actually an enzyme of ketone body synthesis. Alright, so not only cholesterol synthesis, that ketone body synthesis from lipid metabolism is also a vital part in your PG preparation. So keep it in mind, guys. The answer to this will be 3B, HMG-CoA lyase. Moving on, which of the following is the correct sequence of organs involved in vitamin D synthesis? And I think all of you must have answered it correctly. As you know, vitamins are very basic chapter and very important for PG MCQs, and in specifically this case in vitamin D, it starts from the skin under the effect of sunlight from 7-dehydrocholesterol to the first oxidation at the 25th position in the liver, and then the final 1-alpha hydroxylation in the kidney. So the sequence will be skin to liver to kidney. So your answer to this question is 4D. Alright. Moving on. Which of the following enzymes is common to both glycolysis and gluconeogenesis? Now, if you, this is a very easy question. If you remember all the rate limiting enzymes of both glycolysis and gluconeogenesis, you can easily answer this by simply excluding the odd one out. So considering glycolysis, the rate limiting enzymes are hexokinase or glucokinase, phosphofructokinase 1, and pyruvate kinase. So, and these enzymes are not reversible, right? So we rule out option A and option B because these are unique enzymes of glycolysis. Next, we come to the enzymes of gluconeogenesis. So these steps are specifically reversed with the help of enzymes. All right. So the enzyme that reverse hexokinase or glucokinase is glucose 6-phosphatase. The enzyme that reverse phosphofructokinase is glucose 1,6-bisphosphatase, and the enzyme that reverses the action of pyruvate kinase are 2. Okay, pyruvate carboxylase and phosphoenol pyruvate carboxy kinase, that is number 4, the option that is given over here. So A, B and D are unique and reversible enzymes that are not playing role in both the pathways. So the enzymes that are left out, that is number C, phosphoglycerate kinase, it is a reversible enzyme. It converts 1,3-bisphosphoglycerate to 3-phosphoglycerate. Okay, that common step in glycolysis that is also reversed in gluconeogenesis with the help of same enzyme. So this enzyme is common to both glycolysis and gluconeogenesis. So the answer to this question is 5C. Moving on, DNA methylation is not related with. Okay, so DNA methylation is a very important epigenetic process in which a specific cytosine residue, a methyl group is added to the cytosine residue with the help of the enzyme deoxyribonucleotide methyl transferase or DNMT1. All right. Now there are variations of this enzyme. There are other types of DNA methyl transferase. They are DNMT3A or DNMT3B. Those help in de novo DNA methylation. Now all of them, okay, majorly plays a role in masking a DNA. All right, by masking I means protecting or making the DNA identifiable. In case of mismatch repair, you can clearly see that there are residues that are methylated. And again, in case of DNA replication, there are methylation which helps in the identification of the residues, which is the mother strand and which is the daughter strand by the replicase enzyme. And in case of gene silencing too, the de novo DNA methylation takes place and they perform the same mechanism. The intention is same, so that those DNA residues that are methylated are not silenced. All right, so if you read in detail how siRNA etc. are formed, you will see over there that again DNA methylation is happening. So our question is where it is not happening. Okay, so in mismatch repair, DNA replication, as well as in gene silencing, everywhere DNA methylation does take place, but where it doesn't, that is in capping. So 5' capping and poly A tailing, those are post transcriptional modifications of DNA and in which there are no DNA methylation. So answer to this is 6D. Now the next one is an applied question, but it's easy. Which of the following will be an incorrect laboratory finding in RH incompatibility? Now what happens in RH incompatibility? Hemolysis happens. Okay, so due to hemolysis, hemoglobin is released in the plasma, and that hemoglobin will be converted to bilirubin, and that bilirubin is unconjugated. All right, and then the unconjugated bilirubin is converted to urobilin and urobilinogen. All right, urobilinogen, urobilin, all those things. So raised unconjugated bilirubin, true. Remember, our question is which will be an incorrect. Okay, so very important, we need the wrong answer. We need to select the wrong answer. So raised unconjugated bilirubin is absolutely correct. It will happen. Raised stool and urobilin will also happen, because as I told you, the bilirubin is converted to uro and urobilin. Now increase urine bilirubin, that does not happen, because this unconjugated bilirubin is actually not soluble in water, it is insoluble. Increase in urine bilirubin will happen in case of causes of conjugated hyperbilirubinemia like obstructive jaundice, but not in this case. So in case of RH incompatibility, where there are unconjugated hyperbilirubinemia, urobilin will be raised, unconjugated bilirubin will be raised, but not urine bilirubin. So answer to this is 7D. Next, which of the following mediates extracellular transport? Lysosome, exosome, peroxisome and ribosome. Now even if you have not, okay, let me tell you right away, the answer is exosome. All right, these are membrane bound vesicles, extracellular vesicles, mind it, that are consisting of protein, lipid, miRNA, microRNA, DNA and RNA, exosome, and they actually research is undergoing extensively on exosomes, and they have been hypothesized to play a role in intracellular cell transport. Actually they go and they go to the distant cells from their site of origin, and they can empty their entire content, and thus these contents will go inside the cell, and thus they play a role in intracellular transport. Now even if you have not heard of exosomes, this question could have been answered smartly. Lysosomes, peroxisomes and ribosomes, those we all know, right? And we can easily and confidently say that they are actually not involved in intracellular transport. So in this case, the one student who is smart and marked exosome as a diagnosis of exclusion, I will congratulate him or her, because you are right. Moving on to the next question and the last question from biochemistry in this year's Aims exam, June PG exam, ammonia is detoxified in brain as ammonium ion, urea, alanine, and option D, glutamine. So what's the correct answer? Of course, the correct answer is D, glutamine. This is the famous and the most common diagram that you come across in any standard textbook. This is taken from Lehninger, and as you can see, alpha ketoglutarate combines with ammonium ion to reversibly form glutamate. Okay, and then glutamine synthase, again ammonium ion combines with glutamate to form glutamine, and this glutamine is actually finally let go from the brain. So ammonia is detoxified in the brain as glutamine. From the given options, ammonium ion definitely not, because they need to be detoxified. Urea, no, urea is not the answer, because from brain first glutamine is lost. Okay, this is the mechanism. And no, alanine is not the right answer. So remember students, alpha ketoglutarate, glutamate and glutamine. And also you need to remember the mechanism of other mechanisms of ammonia toxicity. As you can see, excess ammonia will lead to depletion of alpha ketoglutarate, and it will lead to hampering of TCA cycle. Glutamate will be excreted, and then GABA will be exchanged, and serotonin, excitatory inhibitor neurotransmitter, and most importantly the new hypothesis, that is NMDA, new N-methyl deaspartate receptors, are one of the mechanism how ammonia is toxic to the brain. So that was it students. This was the 9 questions that came in this year's biochemistry PG examination. So on behalf of biochemistry team of Medeasy and Hippocampus, I feel proud to announce this question, and I'm glad to help you guys. Subscribe to the page. Okay, subscribe to the biochemistry page of Medeasy. More exciting content are coming very soon, which will help you in both undergraduate as well as your postgraduate preparation. I'll leave the link in the description. Feel free to share this video with your colleagues and seniors and juniors who are interested in preparing PG. Get back to me in the comments, and I will see you in the next one. Till then, bye and take care.